ꯑꯩꯈꯣꯏ ꯉꯁꯤꯒꯤ ꯇꯣꯄꯤꯛꯁꯤꯅ pollen pistil interaction haidi class 12 biology chapter 2 sexual reproduction in flowering plants gi topic nebako pollen pistil interaction khoi mamagda agents of pollination tawkhre outbreeding devices tawre huzik ngasi tawduisina pollen pistil interaction ko pollen pistil interaction haibadi stigma da pollen na taraga tabadagi horaga pollen tube na ovary yoba phawgi event to pollen tube sida pollen se pubidoni ko haidi male gamete se pubidoni ba aduga ovary da yoba phawgi event to buneba pollen pistil interaction hai se ko tudi horase ko pollination does not guarantee the transfer of the right type of pollen compatible pollen of the same species as the stigma pollination na haidi pollination ki mathau se kaino pollination je just process amane pollens na stigma da tabagi process haidi transfer tawbagi process to bani ba pollination haise madi pollen se pubi ba khatane ba ko pollination ki process ta ise pollen se stigma phawa pubi ba khatang ne ba ado pollination sina haidi stigma duga chana ba pollen puri bra na wrong type haidi chana da ba pollen puri bra aduga chana ba pollen puri bra haibe se mana pollination sina grantee does not grantee habidi mana daita low de ba ko pollination ki mathau di stigma the pollen pubiba khatane ba ko often pollen of the wrong type either from the other species or from the same plant if it is self incompatible haidi often pollen of the wrong type ko haidi chana da pollen aran ba pollen se ko either from species amadagi oigra natraga same plant chamana ba plant tagi oigra pollen se purak to se ko if it is self incompatible haidi chana da ba do ko ado pollination se granty lo da mana daita pu de ba magi mathau je just pollen se stigma da pubi batu khatane ba ko the pistil has the ability to recognize the pollen whether it is of the right type compatible or of the wrong type incompatible adubu pistil dina ability is pistil na di recognize taw bangam me bako khang bangam me bako pollen se maga chana ba pollen ni natraga compatible or pollen ni natra ma chana da ba pollen ni incompatible or pollen ni habese pistil hadi stigma sina di khang bangam me bako if it is of right type, the pistil accept kaigumba pollen sina ne right type pollen oiraba ko pistil ne accept orani ko the pollen and promote post pollination event ko aduga promote haidi mathangi event to post pollination event haidi mathangi event se chathana bagi damakta ne mana promote mana ayaba pire bako that next paragraph leads to fertilization aduga post pollination haidi post pollination event se mana promote ni makhagi event makha chathal ba yaba event se mana ayaba pirini ko aduga fertilization se haudoi ne ba if the pollen is of the wrong type the pistil reject the pollen by preventing pollen germination on the stigma or the pollen tube growth in the stalk pollination sina wrong type oiba haidi chana daba incompatible oiba pollen oiba ko stigma da haidi stigma da ne pollen se tar su ko haidi pollination se thokraba matung oi tar su ko pistil sina ne haidi stigma duna ne lawsan de reject oiba magi father oiba haidi adugi matung da thokadaba event sing se thokhalaroi ba haidi pollen si ju germinate to halaroi aduga stigma du da ko germinate toraga pollen tube su growth oi halaroi pollen tube growth oi ba di akoi jampor dagi pollen tube se ngasai kai pollen grain gi structure tamba matamda sporopollenin exines sporopollenin same hamna or magi outer membrane se ado jampor hai se sporopollenin absent oi badam jaga du na tro jampor se sporopollenin absent oi ba jaga ni dana ado jampors degi pollen tube se growth tawba horok to ne ba ko aduga style da style haidi pistil gi magi part ahum leidana stigma se pollen tarni style aduga hojik diagram de yarakani ko aduga khagyan tani har ke style aduga ovary ahum leidana adda style da growth tawdini style sum sangtharak toini ba pollen tube se growth tawraga ko jampor de gi ne ba pollen tube growth tawba hawruk to she. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. Sidi adum haidi pistil sina ne pollen se. Pistil sina pollen se reject haidi chana bo ragana lausalani adu chana da bo ragana reject tawba haisidi adum mabani gi marakta continuous adum chatharaga lairaba pot ni bako. This dialogue is mediated by chemical components of the pollen interacted with those of the pistil. Sigi dialogue haidi mana sigumba sathai se continuous hoina chathare ise kamaina no haba matamda pollen te yawba chemical components se pistil se pollen te yawba chemical components se pistil ge interact tor matung da maga chanai natraga chana de sigi pollen si di maga chanai si pollen si ma chana de have se pistils na khangi ne ba kamain khangi no haba matam da chemical components kai de yawba pollen te yawba duna. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by acceptance or rejection. How ki bacha hi khara sidane botanists sing sinane pollen ka pistil ki components se identify tawba ngame aduna mabani gi interaction se haidi rejection bu oiro aduga acceptance bu oiro mabani gi ani sigi pollen pistil haidi interaction se ani sigi interaction se makhoina recognition haidi khangba ngama khibane mabani de yawba components haidi chemical components sing se khangap degi ko identify tawrak bedegi. As mentioned earlier, following compatible pollination, the pollen grain germinate on the stigma to produce pollen tube through one of the germ pores. Aina ngasai haibado. Akoi mamang da hai khredana gaino haidari compatible oraba pollination se. Pollination compatible oi haitari stigma duna pollen se accept tawrabani ko maga chanare na. Aduga tawramnina magi event atopa makha chatharani event se pollen se germinate tawrani. Germinate tawba haidi develop tawba haimne pollen se develop tawraga stigma du develop tawrani aduga pollen tube se form tawrani ba pollen tube se ko pollen grain du degi pollen tube form tawrtisne karai degi jampor degi aina haibado sporopollenin absent oiba jaga ma leibado adudu jampor ni haina jampor degi pollen tube growth tawba horoktoni ba. The contents of the pollen grain move into the next paragraph. Pollen tube. Heidi, the contents of the pollen grain moves into the pollen tube. Heidi, ngasagi pollen grain the yauriba contents. Heidi, mangon the yauriba mayam sing the male gamete, adegi yauriba vegetative generative cells sing the pollen tube the loy move to bahawrani ba. Pollen grain to the yauriba mango de pollen tube che growth tawba hawra ani na pollen aduga pollen tube to the move tawba hawra ani ba. Pollen tube grows through the tissues of the stigma and style and reaches the ovary and hydrogen. Pollen tube se grow tawra ani ne jampor de aduga stigma through the stigma the style se power la ani aduga grow adugi duda reach tawrani aduga ovary de yauna ba chatori ne ba pollen tube sina ko sum la aga ovary de yauna ba chatori ne diagram de garakani diagram de explain tawra gyan tarakani ko. You would recall that in some plants pollen grains are set at two cell condition, a vegetative cell and a generative cell. Akoi mamang da khangkre kaino haide pollen grains se two cell condition su leiba ko vegetative cell habama generative cell nakanda uriba pixel amana vegetative cell ki cytoplasm da generative cell floater leiri se nakanda uriba picture se sisine ko pollen grain ne sise aduga insects plant adugumba plant the generative cell divides and form the two male gametes during the growth of the pollen tube in the stigma adugumba plant karamba plant pollen grain na two cell condition laibado amana vegetative amana generative two cell condition laiba plant sing dadi generative cells amuk divide tawrani na divide tawraga male gamete ani form taweba ko during the growth of pollen tube growth tawringai da ma se divide tawrni aduga male gamete ani form tawni ba ko in plants we set pollen in the three cell condition pollen tubes carry two male gametes from the beginning ko plants lay ba ko three cell condition lay ba plants lay ba ko pollen adugumba plant da di pollen tube se two male gametes carry tawrani dana male gametes ani karamba vegetative cell do divide tawra male gamete ani form tawra manatra ahum sui have manatra three cell haide amana vegetative cell ogedaba two male gametes ne aduga vegetative ne si ahum se ogedaba vegetative cell ahum three cells te side vegetative cells si ahum ani malaya ko asida vegetative cell ga ahum si ne ba mana three cell condition hai se ko adudi yen se ko pollen tube after reaching the ovary enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus pollen tube sinane ovary aurani ko pollen tube se ovary yaunaba puni haba natra ovary aurani ovary auraga mase ovary gi ovule do ovary de yauba ovary manung de yauba ovules de enter tawrni haidi through the micropyle region da ko ama embryo sex se result ani le hadena opposite region ani amana chalaza el amana micropyle en habena tro ama ado micropylum de changani ba ko ma se pollen tube se micropylum de changani and then enters one of the synergid micropyle de yauba ovule de yauba synergids laidana synergids antipodial sujic diagram ulaga yan tarakani ko sina kan de uriba picture se si yung synergids aduga synergids tugi manung da amuk yauba filiform apparatus se fauraga laktoini ba pollen tube se ko many recent studies have shown that filiform apparatus present at the micropylar part of the synergid guide the entry of the pollen tube haidi studies ko haidi filiform apparatus ki mathau se kaino synergids te yauba filiform apparatus ki function se kaim natte ba madi pollen tube se guide tawbiba khataneba guide tawba haidi pollen tube se kadom da chatkani karamba side ta chakani habado mana lambi do takpiba filiform apparatus ki function se ko mathau se si se important ko function of filiform apparatus haidi kaino madi guides the entry of pollen tube ko all these events from pollen deposition on the stigma until pollen tube enter the ovule are together referred to as pollen pistil interaction aina sai ha ahobada haibado pollen pistil interaction haibadi gaino haba matamda pollen sina stigma da deposit tarani aduga pollen tube tuna amuka ovule yaudur phawbagi event to adudu bunyeba pollen pistil interaction haise next as pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. As pointed out earlier, akhoi mamang da point tawkhibado ko pollen pistil interaction is a dynamic process involving pollen recognition. Pollen se recognize tawba khangdok pa machangi ko aduga promotion oigera ma se further event ta promote tawbo oigera haije ma se accept tawbo oigera natraga reject tawbo oigera aduna ma se pollen pistil interaction se dynamic process ni haise adugineba the knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction, even in incompatible pollination to get desired hybrid. The knowledge gained in this area would help the plant breeder ko. Haidi si myam se khang ba sinane plant breeder sing sinane. Kaigumba pollen pistil interaction se compatible oidrasu chanadrasu ne hybrid puthoknaba tongan ba haidi species tongan ba ko cross tawraga hybrid haibedo puthokpado adu tawna ba haidi moida knowledge ama gain taweba ko karmaina hybrid se puthokani haibedo chana da tarsu pollen ga stigma ga se chana da tarsu ko. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, Crotalaria, balsam, vinca on a glass slide containing a drop of sugar solution about 10%. Akoina pollen germinate tawbase ko akoina easily hadi kaidawba yaeba study tawba yai khangmanga yaeba kamaina no haiba matamda pollen khara lawrni ko flower amadagi pollen akoina lay ama samna hairabada lay china rose to aduda akoina magi stigma do paiba matamda hadi anther do paiba matamda akoina khutta yellowish oiba ma paklakpado adu kaim natteba pollen grain ne ba ko adugi pollen grain do glass slide ta lobirani ko dust tobirani aduga sugar solution do drop ama thadarani ko 10% gumba after about 15 to 30 minutes leraga observe the slide under the low power lens of the microscope to low power lens ta akoina observe tawrani you are likely to see pollen tubes coming out of the pollen grain adu kanda akoina ne kamaina kai urakani pollen grain du dagi ne ngasai dust tawkhiba glass slide to the dusting tawkhiba pollen grain du dagi pollen tube se grow tawrapo urakani bako haidi pollen tube to pollen grain gi jump du dagi tholakpado urakani ba khoina. Next, artificial hybridization. Artificial hybridization is one of the major approaches of crop improvement program. Artificial hybridization haise ko crop improvement program makhala maneba ko major approaches haidi khwaidagi chawba approach haidi crop se kamaina crop breeding kamain tawni improve kamain tawni haise khwaidagi hauzik artificial hybridization je hauzik tawnari bane ba ko मैम तौनरी ngata artificial hybridization program se. In such crossing experiment it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. In such crossing adugumba program ko adugumba cross tawbade hybridization haise tongana pollen species samadagi pollen oiro adigi same plant de pollen oiro haidi chana dragasu adu do cross tawbagi experiment makhalam neba ko. It is important to make sure that only the desired pollen grain are used for pollination. Haidi aikoina aina tawre khalase ba hybridization se ko aina pamba pollen du khatane ba desire oi ba aina pamba pollen tu khatane ba pollination thokna ba stigma du da pollination thokna ba tawdori se hadi atopa tongan ba nakan atopa dagi pollination manglak laksan ba do hadi pollen laksala pollination thokpado thokhaloi dori ba aina pamba pollen grain do aina plant amagi pollen grain se pamine adugi pollen grain to stigma do ta hapsalaga pollination thokhandune adudune ba artificial hybridization to hon bhai se is protected from contamination stigma do da pollen grain se tahandoi natro stigma si sum haidi exposed or ready pollen se amadagi amuk haidi agents yamanina bird no oiro bee no oiro butterfly no oiro mayam na purakani kana wind no oiro adu stigma do contaminate tawdana ba protect tawdoine ba haidi stigma do ko adu kamaina protect tawbono yengse ba unwanted adu su palm daba pollen taki aina pamba pollen to khatane ba pollination thokhandori se adu do kai na tawhale habada emasculation ga bagging technique na tawhale dudi yengse ko emasculation hais na kai no habse If the female parents bear bisexual flower, removal of anther from the flower, but before the anther dehisce, using a pair of forceps is necessary ko. Kaigumba female parents bear ko. Female parent to na ne bisexual flower habidi kaine ba male flower su female flower su pulap pansan bado ko yaushan bado aduna anther se adugi anther do remove tawbam changye ba ko anther do da ka yaw natro pollen grain yawi natro flower do gi anther do remove tawbam changye ba ko before the anther dehisce already pollen grain mangon da tholak to pollen anther da gi thok to ne dehisce already ada using a pair of forceps is necessary ad forceps sizinaraga ne lokthokpam changye ba ko flower do dagi nakanda uri ba pic se sisine forceps taraga lawthu ine ba ko and third this step is referred to as emasculation haidi adu dubune ba haidi bisexual flower madagi the anther do dehisce tawdringai da pair of using a pair of forceps ka lawthok pod anther do lawthok remove taw pod adu dubune ba emasculation haise. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. Bagging haise na kaino ngasai emasculated flower di anther remove tawkraba. Anther do forceps ka taw remove tawkhre ko adugi remove tawraga remove tawkhibado bag amana ko haidi butter paper bag amana cover tawsanba ma butter paper gi oiba ko bag amana plastic haigera ko bag amana suitable size ki ko chana ba magi size tu gi chana ba de cover tawsandoine kai gi no haba matam da stigma do atop pollen tasalaktana ba mangonda haidi atop plant ki mana pamda ba pollen to tasalaktana ba hana cover ta hodoine ba ko cover ta budu bene bagging haise nakanda uriba pixine ko haidi ngasagi emasculated flower do magi stigma do contaminate tawkhidana ba bag amana cover tawdone ba ko butter paper gi black bag oigera ko when the stigma of bag flower attains receptivity ngasagi flower haidi flower do gi stigma do bag flower haidi ngasagi cover tawsan khibas flower do gi stigma do na ne receptivity si attain tawra aba haidi mase mana pollen se lawsan ba yare mase matam chap chare haiba matam dane mature pollen grain se collect tawrani ko atop anther of the male parent dagi akoina haidi mina ni ko collect tawdori se agents of pollination sijina dauna te win sijina dauna te bee butterfly amta sijina dauna te akoi mina tawdoba mina collect tawrani dana atop male parent na badagi the state on the stick ba odu the stigma adu the dust tawdoni ba khoina adum haidi chaishandoine ba stigma aduda aduga flower do amuk rebag tawbira flower do ngasai cover tawbudu maina ne amuk cover tawbirani bag to da and the fruits allow to develop aduga fruits do mahai yalaktre phok thamdone ba next if the female parent produces unisexual flowers female parents tuna unisexual flower oirba there is no need for emasculation female parent unisexual oiba haiter di male flower khatang oigera female flower khatang oigera ama khatang yaubodaba flower du da adu idi emasculation tawdrasu yaiba emasculation haidi animak yausal bisexual oira kandani emasculation tawrisidi ko the female flower buds are back female flower buds to adum bag tawbirani bagging di tawni ko haidi atopagi pollen unwanted oiba pollen to pamdaba pollen to tasalaktana ba before the flower open flower duna expose tawdringai satoraktringai da when the stigma becomes receptive stigma sina mature oirakandane pollination process se thokhalani using desired pollen and the flower rebag adudi bagging gi process se chap manarni ko emasculation tawde haiba khatane ba unisexual flower ri bagging gi process chap manarni stigma do receptive oima matamda pollen to apamba pollen to akhona dust tawbirani aduga flower do muka rebag tawbirani ma mahiyandri phawba ko ꯍꯥꯏꯗꯤ ꯀꯣꯚꯔ ꯇꯧꯕꯤꯔꯅꯤ ꯉꯁꯥꯏ ꯕꯇꯔ ꯄꯦꯄꯔꯗꯨꯅ ꯑꯗꯨꯒ ꯉꯁꯥꯏꯒꯤ ꯗꯥꯏꯒ꯭ꯔꯥꯝꯁꯤꯡꯗꯣ ꯒ꯭ꯌꯥꯟ ꯇꯥꯅꯕ ꯑꯝꯇ ꯌꯦꯡꯁꯦꯀꯣ ꯗꯥꯏꯒ꯭ꯔꯥꯝꯗꯣ ꯑꯩ ꯑꯦꯛꯁꯄ꯭ꯂꯦꯟ ꯇꯧꯒꯦꯕ ꯗꯥꯏꯒ꯭ꯔꯥꯝꯁꯦ ꯌꯦꯡꯁꯦꯀꯣ ꯗꯥꯏꯒ꯭ꯔꯥꯝꯁꯦꯀꯣ ꯉꯁꯥꯏ ꯑꯩꯅ ꯍꯥꯏꯕꯗꯣ ꯅꯥꯀꯟꯗ ꯑꯩꯅ ꯄꯤꯛ ꯑꯃ ꯄꯤꯔꯤꯁꯤꯁꯨ ꯄꯣꯂꯦꯟ ꯒ꯭ꯔꯦꯟꯗꯣ ꯁ꯭ꯇꯤꯛꯃꯥꯗ ꯇꯥꯔꯛꯑꯅꯤ ꯑꯗꯨꯒ ꯁ꯭ꯇꯤꯛꯃꯥꯅ ꯂꯧꯁꯟꯕꯒꯤ ꯄ꯭ꯔꯣꯁꯦꯁꯇꯨꯅꯦꯕ ꯑꯩꯅ ꯄꯤꯛ ꯎꯠꯂꯤꯁꯦꯀꯣ ꯑꯗꯨꯒ ꯂꯥꯏꯔꯤꯛꯇ ꯌꯥꯎꯕ ꯁꯤꯒꯤ ꯗꯥꯏꯒ꯭ꯔꯥꯝꯁꯤꯗꯣ ꯑꯦꯛꯁꯄ꯭ꯂꯦꯟ ꯇꯧꯒꯦꯀꯣ ꯁꯤꯅꯦ sida pollen grains tareko pollen grains germinate tawre man receive tawbe germinate tawraga si pollen tube growth tawrise si pollen tube ne ba si se man sida thai se pollen tube ne ado ina isda si sina stigma si pollen grain tapham se stigma ne ba stigma ko stigma aduga sina style haibedo style da growth tawi haida style power chatoi ne ba ko si style aduga ovary yaudori ne ba sina ovary ovary ko sina ovary ovary da muka ovary gi manungi ovule sida changdo embryo sac natra se embryo sac lere ko si ovule se changdori ne ba si ovule sida aduda yangoto synergids lere egg cell lere polar nuclei lere aina mamangda explain phajana tawkhamni ko yengdaba yawra so dum video gi part sing to aina link ta dum hapake aduga yengo ko aina si explain am tawkhrabani embryo sac ki amanung haidi inner embryo sac ta yawriba cell sing to egg cell synergids sing se kai kai yawe habese aina loin explain tawkhrabne ba ko lere pollen tube to sineba pollen tube se sineba ko pollen tube se sine pollen tube si macha se pollen tube neba ko pollen tube sida pura ise male gametes ne vegetative nucleus ne vegetative nucleus hadi vegetative cell du neba ko 3 cc ki se ma se 3 cell condition ne sigi sina ko aduda si synergids ne ko synergids lere synergids manung da filiform apparatus layer filiform apparatus ne guide to be hydra pollen tube se adu guide to biri ne duga egg nucleus layer central cell layer na polar nuclei male gametes double fertilization haise kaino haibese pollen pistil interaction gi matung da haidi akoi double fertilization tawdene topic se aduna next video da ko aduga si amuka hena gyan tarani ko si se male gametes sing do pollen tube to yausalle synergids te yauba filiform apparatus na guide tawbire aduda mangonda discharge tawfre male gametes te ko male gametes amana synergids change amana egg nucleus ka fuse tawdone ba ko haidi egg apparatus to egg nucleus ka fuse tawdone ba ado double fertilization da hena gyan ta na ba ai explain tawrage double fertilization do na ini video si am sangani na double fertilization do next video gi oina thamme ko aduna gyan ta daba kok ngau ba haidi gyan ta daba lairasu doubt level lairasu comment adum hairo o ai adum clear tarake video se like subscribe share tabio thank you